Today, I am taking up the topic sex determination in animals. There was a lot of speculation about sex determination in human beings and other animals for a long time. And it was a puzzle how sex is determined, how some are born as males and why some are born as females. For the first time, in the year 1891, a scientist called Henking was studying spermatogenesis in insects. Then he found that 50% of the sperms received a specific structure and other 50% did not receive this specific structure. He couldn't understand why only 50% of the sperms receive this, and he called it as X body. Later on, scientists have found that the X body is nothing but the X chromosome. Later on, other sex chromosome, Y, is also discovered. So, in human beings, the sex determination is due to the sex chromosomes X and Y. Then, two scientists, namely Stevens and Wilson, proposed the chromosomal basis of sex determination. According to them, sex is determined by the sex chromosomes. Then, in our cells, we got chromosomes. These chromosomes are two types, autosomes and allosomes. Autosomes are also known as somatic chromosomes, and they regulate all bodily functions other than sex determination. The allosomes or sex chromosomes are mainly concerned with sex determination. They decide the sex of the organism. Then, there are various methods of sex determination in animals. They are known as XXXY type, found in mammals, including man, and some insects, including Drosophila. XXXO type, it is found in some insects. ZZ type is found in some in the birds, reptiles, and some fishes. ZZ type, it is found in moths and butterflies. Haplodiploidy, it is seen in the honeybee. Then there is a very interesting theory known as genic balance theory. It was proposed by a scientist known as Bridges in the insect Drosophila. Then, here we have to remember two terms, homogametic and heterogametic. If an animal produces gametes of two types, it is known as heterogametic. If an animal produces only one type of gamete, it is known as homogametic. Let us see how this happens in different animals. First, let us take up XXXY type. So, in human beings, Drosophila, other mammals, and some insects, we come across this type of sex determination. As the autosomes are not involved in sex determination here, I have not drawn about the autosomes. I have depicted only sex chromosomes. So, females. In human beings, females have two X chromosomes. Males have one X and one Y chromosome. So, females produce only one type of gamete, and that carries the X chromosome. So, females are homogametic. Coming to males, they got X and Y chromosomes. 50% of the sperms receive this chromosome and other 50 receive the Y chromosome. So, males are heterogametic. Then, during fertilization, if this egg is fertilized by this sperm, we get XX here. So, it develops into a female. If this egg is fertilized by this sperm having the Y chromosome, it develops into a male. So, in this way, sex is decided in human beings and Drosophila and other insects. The second one, XXXO type. This type of determination, sex determination, is found in some insects. which was studied by Henking. So here, the female insects are having sex chromosomes XX, and male insects are having the sex chromosomes only one X. 0 means there is no second sex chromosome. So males have one X chromosome, females have two X chromosomes. So the females, they produce only one type of gamete, and that gamete carries X chromosome. So females are homogametic here. Here what happens is only one X chromosome is present. So 50% of the sperms receive the sex chromosome, and other 50 do not receive any sex chromosome. So that was studied by Henking. So 50% receive the sex chromosome, and other 50 do not receive that. That means male is heterogametic. Here, if this egg is fertilized by the sperm, we get XX chromosomes, and it develops into a female insect. If the egg is fertilized by the sperm carrying no X chromosome, it has only one X chromosome and develops into a male. In this way, in some insects, sex is determined. Then the third type, ZZ type. This is seen in birds, reptiles, and some fishes also. Here you see, this is the female, this is the male. In females, the sex chromosomes are represented with the letters Z and W. That means these two sex chromosomes are different. So when during gametogenesis, what happens? This Z chromosome enters one egg and W chromosome enters another egg. That means the females produce two types of eggs or gametes. So female is heterogametic here. Remember very carefully. In the birds, reptiles, and fishes, females are heterogametic. Coming to the males, males have two Z chromosomes, and all the sperms produced by them receive one Z chromosome. So males are homogametic. Then, if this egg is fertilized by the sperm, we get ZZ combination, and it develops into a male animal. If this egg is fertilized by the sperm, we get ZW combination, and it develops into a female. In this way, in the birds, reptiles, and fishes, sex is decided. Then the fourth type, ZO, ZZ type. It is found in moths and butterflies. These are insects. In the insects, moths and butterflies, we come across ZO, ZZ type. See the female here. Female has both the ZZ chromosomes. Males have ZO chromosome. So, females produce only one type of gamete, so they are homogametic. Males produce two types of gametes because they have only one chromosome Z. So males are heterogametic. So here, 50% of the sperms receive the Z chromosome, and other 50 do not receive any sex chromosome. Then, if this egg is fertilized by this sperm, we get a combination of ZZ chromosomes, and it develops into a female. If this egg is fertilized by this sperm, it receives only one sex chromosome, and it develops into a male. In this way, in moths and butterflies, ZO, ZZ type of sex determination is seen. Then, there is one very interesting type of sex determination in the honeybee. This is also seen in some of the ants and other insects. This is also known as hymenopteran type. Here, the queen bee has 32 chromosomes. The males or drones have only 16 chromosomes. That means the queen bee, which is a female, is having diploid number of chromosomes, whereas the males or drones have only haploid number of chromosomes. Here diploid number means 32, full quota. Haploid means only half, that is 16. That's why it is known as haplodiploid mechanism. Here what happens is, the female produces eggs through meiotic division. The queen bee produces eggs through meiotic division, and the eggs receive 16 chromosomes each. Coming to the males or the drones, already they have haploid number of chromosomes. So they cannot exhibit any meiotic division. So the interesting thing is, during gametogenesis of drones or males, mitotic division takes place. In all other animals, meiotic division takes place during gametogenesis, but here mitotic division takes place. That means there is no reduction of chromosomes. And all the sperms receive 16 chromosomes. So here, the females are producing eggs with 16 chromosomes, and the males are producing sperms with 16 chromosomes. But the difference already I have told you. In the case of females, it is meiotic division here during gametogenesis. In the case of males, during spermatogenesis, it is mitotic division. Then, some eggs are fertilized by the sperm. If the eggs are fertilized by the sperm, what happens? The zygotes receive 32 chromosomes. That means they are in a diploid state. And these eggs, fertilized eggs or zygotes, develop into females. These females, some of them develop into queen bees and others develop into worker bees. So, in all the female honeybees, the chromosomal number is diploid, that is 32. And they develop from fertilized eggs. The queen bee also produces some unfertilized eggs. Here there is no fertilization. These eggs, unfertilized eggs, also develop. When an unfertilized egg develops, it is known as parthenogenesis. It is known as parthenogenesis. So the unfertilized eggs, which are haploid in number, they develop into males or drone bees. That's why in the male bees, the chromosomes are only haploid or only half. That's why this mechanism is known as haplodiploid. Males have haploid, females have diploid number of chromosomes. This is about haplodiploidy, unique to honeybees. The last one is known as genic balance theory. It was proposed by Bridges, and for this he received the Nobel Prize. Till now, we spoke about the role of sex chromosomes in the determination of sex. But this theory appears to be very odd. According to Bridges, not only the sex chromosomes, but also the autosomes contribute to the determination of sex. Here sex chromosomes means only X chromosome. In Drosophila, according to him, Y chromosome is required only for fertility. Except for that, it has no role in determination of sex. So, according to Bridges, in Drosophila, both autosomes and X chromosomes participate in sex determination. Here, autosomal sets are considered. A means autosomal set. X means X chromosome. Here, the ratio between the number of X chromosomes and the number of autosomal sets is important. Autosomal sets is important. So, if the ratio between the number of X chromosomes and autosomal sets is 1, that zygote develops into a female. If the ratio between the number of X chromosomes and autosomal sets is 0.5, it develops into a male. If the ratio is between 0.5 and 1, then the zygotes develop into intersexes and sterile insects. They are neither males nor females but sterile. If the ratio is less than 0.5, the zygotes develop into meta males or super males. If the ratio is more than 1, they develop into super females or meta females. Remember, these super males, super females, they are fertile. So, these are various methods of sex determination in animals. Thank you.